Imagine your body as a fortress under siege, but instead of soldiers holding the gates, you have millions of microscopic factories humming inside every cell. That's not science fiction. That's what happens when we teach our immune system to build custom weapons on demand. Today, we're diving deep into the revolutionary discovery where B cells transform from passive defenders into active surveillance machines, pumping out tailored antibodies to hunt down cancer and HIV with surgical precision. This isn't just about surviving an infection anymore. It's about turning your own biology into a targeted eradication engine. By the end of this video, you'll understand how scientists are rewriting the rulebook for treating once incurable diseases and why this shift means the end of traditional chemotherapy as we know it. The concept of antibodies is often simplified in textbooks to Y-shaped proteins floating around looking for germs, but the reality is far more dynamic. Think of each antibody as a unique lock that fits only one specific key, which happens to be an antigen like a virus or a tumor marker. When a standard infection hits, your body produces generic copies of these antibodies to mop up pathogens. But what if you could program them? This is where the breakthrough comes in. Scientists have discovered a way to reprogram B cells, the very factories that make antibodies, to become specialized production units. Instead of waiting for an automatic immune response, researchers can guide these cells to produce specific molecules designed to destroy cancer cells or fight HIV directly. It's like switching from building generic fire extinguishers to custom-built drones equipped with pinpoint targeting systems. The specificity here is absolute. One antibody binds to a single antigen with high fidelity, ensuring that the weapon hits the target and spares the rest of your body. This level of precision eliminates the collateral damage often seen in traditional treatments, offering a new paradigm where therapy becomes as surgical as it is effective. So how do we actually turn these cells into active surveillance machines? It starts with the biology of B cells themselves. Normally, they wake up only when they encounter an enemy antigen and then differentiate into plasma cells to pour out antibodies. The new approach bypasses this slow, reactive timeline by directly engineering the genetic instructions within these cells. Scientists have shown that it's possible to coax B cells into becoming factories that churn out specially designed antibodies in high volume without needing a viral trigger first. These engineered cells act as living bioreactors inside your bloodstream, constantly scanning for the specific disease markers we programmed them to look for. Once they spot their target, whether it's a mutated protein on a cancer cell or a viral spike, they release their payload instantly. This turns every B cell into a tiny, mobile missile system that can navigate the complex environment of human tissue and deliver its cargo right where it counts. The result is a self-sustaining defense network that doesn't just react to illness, but proactively hunts it down before it spreads, effectively giving you an internal army that never sleeps and never gets tired. The implications for diseases like cancer and HIV are staggering because both have historically been considered the hardest battles in medicine. For decades, we've struggled with tumors that evolve resistance to drugs or viruses that hide within our own cells. With this new method of antibody production, those limitations vanish. Take cancer. Instead of killing every cell in a tumor indiscriminately, these customized antibodies can bind to specific receptors on cancer cells and flag them for destruction by the immune system itself. The body does the heavy lifting once it's been given the right tool. For HIV, which hides in sanctuary sites like reservoirs within resting CD4 plus T cells, traditional antiretrovirals struggle because they don't kill the virus directly or clear out the infected cells efficiently. But imagine an antibody designed to specifically recognize and dismantle those hidden viral sanctuaries. It would be able to strip away the protective layers that viruses use to evade detection. This shifts the strategy from suppression to eradication. We're moving past a world of lifelong medication management into one where we can potentially clear infections entirely, turning chronic conditions into finite challenges that our biology is finally equipped to solve on its own. What does this mean for the future? It means the pharmaceutical industry and medical research are shifting gears toward personalized immunotherapy at an unprecedented scale. We're no longer just waiting for vaccines or drugs to arrive. We're building living medicines that adapt to our unique biological landscape. In practical terms, this could lead to treatments that require no lifelong dosing, eliminating the side effects of chemo and radiation while drastically improving quality of life for patients. It also opens the door for new types of diagnostics where your body monitors its own health in real time, alerting doctors to disease onset before symptoms appear. The technology is already showing promise in early trials, with scientists successfully creating antibody factories that target specific cancer markers and viral proteins. As we move forward, expect to see these techniques integrated into routine oncology and infectious disease protocols, making them accessible beyond the lab bench. This isn't just a scientific curiosity. It's the foundation of a new era where our bodies are not just victims of illness, but active participants in their own healing process. The future of medicine is here, and it's being built inside your cells right now.